of it in terms of how people communicate every day. It's a daily sort of problem. One of the things that I'm working on now, it's sort of in the sort of user testing stage, is an app. It's a very limited app for the moderators of online discussion forums for museums across the country, science museums across the country. They want to promote science learning on these online forums. And what they want to be able to do is be able to analyze what's going on there. So I've developed with a team of other faculty and grad students and undergrads an app that will take in raw natural language data and then be able to sort of have a low resolution view of what is going on rhetorically in terms of what we call moves, different operations and games that people are playing with language. We take it in on this sort of web app. It outputs this sort of classification schema here, and we have 3 different rhetorical moves. For staging is sort of a move of description, of laying out different factual cases. Inviting is an invitation for conversation, collaboration, inviting some kind of emotional investment, and also evoking a sort of spurring, compelling sort of move that people do in these online discussion forums. So this is what I sort of mean by computational rhetoric in one small portion. Now, as I said, I don't really have a whole lot of data needs. A lot of the data that I work with here and will continue to work with, and some of them are fairly large, but large for me. Not going to be large for any high-performance computing center. So what this means for me in my research and other people in my field will probably mean that we can make things a lot quicker, we can expand our efforts, but really, to me, the main thing that gets me excited about this particular endeavor is this notion that we can transform the humanities, this notion that we can ask new questions in the arts that we have not been asking enough of because we typically do what we call close readings. We have a small sampling of text, small, and we try to derive global understandings of culture, of history, of large-scale epics in human behavior based on these very close readings, these sort of micro readings of text. And what this can mean for us is that, we get these what we call sort of ideational insights. We understand motivation, we understand different maybe trajectories of thought. But now we have the opportunity to correlate our understandings of language with larger scale phenomena that are non-linguistic in many different cases, that are about other types of behavioral data that we have not been, as a field, I'm talking about rhetoric, maybe English, not been very good about incorporating into our methodologies. So what I can see here is this, and I'll break this down into much more simpler sort of hope for me, is that a center, because it is a center, it will bring together hundreds, thousands of very, very smart people. And when you have a collection of very smart people, you can collaborate with very smart people. And for someone like me, having this resource to be able to partner up with people who are outside of my field and have interdisciplinary questions means a whole lot, because we can transform what we normally research. We can incorporate new things that we haven't thought of, and we can learn to ask questions based on people from oceanography, from the life sciences, from all of these different fields that we don't necessarily have access to. And this center is what's going to actually or any type of center or any type of sort of locus will help us ask these different questions that can transform not only my field, but I think many other fields in the arts and humanities. Thank you.
Up. Thank you, Ryan. Is Lenny Moise. He's an assistant professor. Do I have the? Okay. Lenny Moise, assistant professor of research, the College of the Environment, Life Sciences, and Cell and Molecular Biology. Part of Andy DeGroot's group.
Hello everyone, Lenny Moise. I'm part of the CMB department and at the Institute for Immunology and Informatics which is located in Providence. So you probably don't see me down here much. I like coming down here, it's beautiful, and thank you for organizing this wonderful symposium today. So I like to say that good news should travel at the speed of sound and bad news at the speed of light. Unfortunately, this is a lightning talk, so I'm going to try and put the best spin I can on what we do at iCubed. There we are 6 faculty members, and where our mission is to improve human and animal health by applying the power of immunomics to design vaccines, diagnostics, and immunotherapeutics. That is our focus, and we develop new knowledge by engaging in basic and applied immunology research. Our vision is to become a global center for immunology and vaccine research, and to improve the international recognition of URI as a generator of important scientific contributions, and by contributing to the reputation of URI and attracting talent and improving URI's national standing. So what we all do at iCubed is to study immune responses to infection and vaccination. So we spend a lot of time focusing on the pathogen, and as you know, there are haystacks upon haystacks of sequence information that have been generated through the power of genomics. But we don't only think about the pathogen because it interacts with the human host. And so what we're really looking at when we do sequence analysis is to identify what goes on in that interaction at the interface of the two. And in case you didn't know yet, we are really superorganisms. We are, or better known as a bag of bacteria. We're composed of all kinds of these so-called friends with benefits. They're called the human microbiome, and they too interface with us and with pathogens. So immune responses have to take into account coexistence of the host and the human microbiome, and what effect the pathogen would have on it. So what I have been focusing on, as well as my close collaborator Andy DeGroot, is this interaction. Not everyone here is in biology, and not everyone in science, but the important thing to see in this picture here is that we're looking at the interface of 2 cells. And these are the proteins in blue and in green that are at that interface. So this is the information presenting cell, and this is the cell that processes that information and turns it into action, the kind of action that results in protective immunity from that protects us from disease, from infection, from cancer, and from autoimmunity, from attacking our own cells. The message is this red piece here. And we think we're really cool when we send out messages using Twitter, limited to 140 characters. Well, nature has done much better than that. This message comes in the form of 9 characters. Okay? And it leads to those awesome biological effects. Well, we've been looking at the 2 faces of this 9-character message. The side that faces the information presenting cell, and the side that faces the information processing and action cell. For close to 20 years now, people have perfected algorithms that identify patterns of these 9-character messages that will interface on the information processing side, presenting side. What we're working on now in the frontier of this field is finding the patterns that are presented to the action side, and that's really important because some of them may be shared between humans and pathogens, and being able to identify those that are could enable us to design better vaccines and better immunotherapeutics. So this is actually an embarrassingly parallel problem that would be ideally suited for modern computing architecture such as GPU processors, and that is the kind of computing need that I think that we as immunologists are thinking about for future work. But aside from simple sequence analysis, I also see that in the future we will want to do structural modeling of this protein-protein interaction, which would become a lot more complex and require greater computing power. Okay, so that's just the interaction, but I'll tell you the other members of IQ are, as well as myself, are studying what happens downstream on the action side. Once the information is passed on to a so-called T cell, what actually happens to the T cell. So we like to understand the phenotype of the cell, and a single cell analysis approach we use is to describe flow cytometry, with which we can analyze a number of parameters that characterize the cell. So the data that comes from a single cell, from a study of 2 parameters is shown here, 3 parameters here. It gets more complex as you go up to as many as 9 parameters, which is about where we are at iCube today. It's already pretty complex.